Welcome to Sleepy Stories. In this episode, we will take a clear and steady look at the molecule that defines every living thing, DNA. The goal is to help you learn while you rest. If you do not fall asleep right away, that is completely fine. In the time you are awake, you will discover how this elegant thread of chemistry carries the memory of life itself. Simply listen, let your breathing slow, and allow the information to settle at its own pace. Deoxyribonucleic acid, or DNA, is more than a chemical compound. It is a data system built by evolution to copy, store, and transmit information through time. Each cell in your body holds roughly 2 meters of this molecule, coiled and folded inside a microscopic nucleus. Those long strands contain instructions that determine how cells grow, divide, and interact. Every heartbeat, every glance, and every breath rely on the continuous reading of this ancient code. DNA is not alive by itself, yet nothing living exists without it. The story of DNA is also the story of discovery. For most of human history, heredity was a mystery explained by resemblance and chance. Families noticed that children looked like their parents, that crops bred for flavor kept that trait, and that diseases sometimes repeated down generations. But what carried those patterns? Was it blood, spirit, or something invisible hidden in the cell? The search began in the 19th century when scientists first glimpsed thread-like structures under primitive microscopes and suspected they might hold the secret of inheritance. By the early 20th century, chemistry had revealed the building blocks of cells: proteins, sugars, and nucleic acids. Proteins seemed the obvious choice to store genetic information. They were complex and varied. DNA, by contrast, looked too simple: only 4 chemical bases, repeated endlessly. Yet simplicity can conceal power. In the decades that followed, a series of careful experiments showed that DNA, not protein, was the true messenger between generations. It was a quiet revolution, one that shifted biology from observation to decoding. At its heart, DNA is a long polymer made of repeating units called nucleotides. Each nucleotide contains a sugar, a phosphate, and one of 4 bases: adenine, thymine, cytosine, and guanine. The order of these bases forms sequences, and those sequences form genes. The sequence of bases in a gene tells a cell which protein to make and when. A gene does not act alone. Thousands cooperate in networks that build tissues and maintain balance. The beauty lies in the logic, a 4-letter alphabet that can write instructions for every organism that ever lived. When you imagine DNA, picture not a rigid ladder, but a flexible spiral staircase twisting through space. Its paired strands face each other like mirror pages, one reflecting the other through complementary base pairing, A with T, C with G. This design allows perfect copying. When the strands separate, each provides the template for a new partner. Stability through symmetry, renewal through replication, a principle simple enough for chemistry, profound enough for life. As this story unfolds, you will see how DNA replicates, mutates, evolves, and remembers. You will learn how cells read it, how species change through it, and how humanity has learned to read and even edit it. The tone remains calm, steady, and factual, allowing your mind to wander safely between curiosity and rest. Every cell within you is quietly performing the acts this molecule describes, following a script billions of years old, written in invisible letters of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Tonight we will trace that script from its first discovery to its future possibilities. Beneath it all is one simple truth: life is information made stable. And within that stability, every heartbeat writes another line of continuity. Long before the double helix became an icon, The idea that life's traits could be written and passed along was only a suspicion. In the mid-19th century, Gregor Mendel, an Augustinian monk, studied thousands of pea plants and noticed that traits followed mathematical ratios from one generation to the next. His quiet work in a monastery garden became the first hint that heredity followed rules. Yet even he did not know what carried those instructions. His results lay forgotten for decades, waiting for a world ready to understand them. By the early 20th century, microscopes had grown powerful enough to reveal thread-like bodies inside cells, chromosomes. They appeared to divide and rejoin during reproduction, behaving just as Mendel's hidden factors should. Scientists began to suspect that the essence of inheritance lay in these structures. But what were they made of? Proteins, with their 20 diverse amino acids, seemed capable of storing vast complexity. DNA, discovered earlier by Friedrich Miescher, in the substance he called nuclein, appeared far too repetitive, too monotonous to hold the richness of living forms. The 1,920 seconds and 1,930 seconds were a time of speculation and rivalry. Some chemists proposed that genes might be tiny protein knots. Others imagined complex carbohydrates carrying codes of energy. Few considered DNA more than a structural scaffold. Yet one by one, experiments began to shift that belief. In 1928, Frederick Griffith performed his famous experiment with 2 strains of Streptococcus pneumoniae, one virulent, one harmless. When he killed the virulent bacteria and mixed their remains with the harmless strain, the latter became deadly. Something in the dead cells had transferred the power of infection, a transforming principle. Years later, in 1944, Oswald Avery and his colleagues, Colin MacLeod and Macklin McCarty, purified that transforming substance and demonstrated that it was DNA. When they destroyed the DNA, transformation ceased. When they preserved it, transformation continued. Their meticulous controls left little doubt. Still, skepticism persisted. Many biologists clung to proteins as the likely genetic material. Only when Alfred Hershey and Martha Chase used viruses that infect bacteria, labeling their DNA with radioactive phosphorus and their proteins with sulfur, did the argument end. The experiment showed that only the viral DNA entered the host cell and guided reproduction. The case was settled. The genetic instructions resided in DNA. Those findings marked a quiet but profound turning point in science. Heredity was no longer a mystical property of life but a chemical one. For the first time, information itself was seen as a physical entity, carried in molecules that could be isolated, measured, and even weighed. The concept of information had not yet fully entered biology, but the seeds were planted, that life could be read like a text written in matter. Reflecting on that moment, one can imagine the awe felt in those early laboratories. The scientists were not seeking grandeur. They were tracing invisible causes through the lens of a microscope and the curve of a graph. Yet their results would change everything: medicine, agriculture, identity, and the very way humanity understood itself. The mystery of resemblance was becoming an equation of molecules, and nature was revealing that memory could exist within structure. Every discovery built on another, forming a chain as orderly as DNA itself. Griffith's mice, Avery's flasks, and Hershey and Chase's viral phages were like successive sentences in the unfolding story of biology. From them emerged the realization that life's continuity rests on chemistry so subtle it can fit inside a single cell, yet so powerful it can shape entire species. By the early 1950 seconds, the world of biology was approaching a revelation. Scientists knew that DNA carried genetic information, but not how it did so. To understand heredity, they needed to see the molecule's architecture. Its structure, many believed, would reveal its function. a principle already proven across biology. In Cambridge, England, two young researchers, James Watson and Francis Crick, pursued the problem with restless imagination. In London, at King's College, Rosalind Franklin and Maurice Wilkins were using X-ray diffraction to look directly into the invisible world of molecules. Their methods, though different, aimed at the same goal: to visualize the code of life. Franklin's images were crucial. Her meticulous preparation of DNA fibers produced a diffraction pattern known as photograph 51, a diamond-shaped cross that revealed a repeating helical form. Each layer of reflection represented a turn of the helix, spaced at precise intervals. From those measurements, one could calculate the dimensions of the spiral, its width, its pitch, and even the angle between its base pairs. Watson and Crick, studying her unpublished data through intermediaries, realized that the molecule could not be a single chain or a triple spiral as some had proposed, but two strands coiled together in opposite directions. The insight was as elegant as it was simple. Each strand's backbone consisted of alternating sugar and phosphate groups, while the interior rungs were pairs of nitrogenous bases, adenine with thymine, cytosine with guanine. Hydrogen bonds held the pairs together, weak enough to allow separation but strong enough to maintain alignment. This complementarity explained replication. When the strands divided, each served as a template for its partner. Information could now copy itself with near-perfect fidelity, the chemical basis of inheritance finally laid bare. The model took shape not in a high-tech lab, but in the modest rooms of the Cavendish laboratory. Wooden rods, metal plates, and cardboard cutouts were arranged to test possible geometries. Every correct distance and angle produced a visual harmony that mirrored the molecule's reality. When the pieces finally aligned, Watson and Crick saw it, the double helix twisting with grace and logic. In April of 1953, they published their discovery in Nature, writing the understated line that would echo through science. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That sentence changed biology forever. For the first time, heredity was not only observable but imaginable, a ladder of chemistry reaching through time. The structure made sense of everything discovered before: Griffith's transformation, Avery's DNA, Hershey and Chase's viral results. Each mystery clicked into place. The double helix explained how stability and flexibility could coexist, how life preserved information yet allowed change, how evolution had built a memory that could rewrite itself. Rosalind Franklin, whose data made the model possible, continued her research into the structure of viruses, but she would not live to see her full recognition. Her work, later acknowledged as essential, remains a reminder that discovery is often shared among many minds, not owned by one. The beauty of the helix belongs to all who glimpsed it, whether through data, imagination, or persistence. When you picture the double helix, imagine two ribbons winding gently around a common axis, like partners in an endless dance. Each twist measures about 10 base pairs, each turn identical to the last, continuing for millions of steps. It is a design at once efficient and poetic, symmetry holding information steady across generations. Within that shape lies the logic of life, a molecule capable of both memory and change, stability and evolution. And from this simple spiral, all biology unfolds. Every book has an alphabet, and every language has rules that give it meaning. Life, too, is written in an alphabet, one with only 4 letters. These are the 4 bases of DNA: adenine, thymine, cytosine, and guanine. Each one is a small organic molecule, a combination of carbon, hydrogen, nitrogen, and oxygen atoms arranged with delicate precision. 2 of them, adenine and guanine, belong to a group called purines, larger molecules with double ring structures. The other 2, thymine and cytosine, are pyrimidines, smaller and single-ringed. Their sizes and shapes determine how they pair, ensuring the perfect geometry of the double helix. Adenine always pairs with thymine, and cytosine always pairs with guanine, a rule that has never been broken in any known form of life. It may seem almost magical that so little variety can hold so much information, yet the secret lies not in the letters themselves, but in their sequence. Just as the same 26 letters of the English alphabet can produce infinite words and stories, DNA's 4 bases can form endless combinations, writing the instructions for everything from bacteria to humans. The genome of a simple virus may contain only a few thousand bases, that of a human over 3 billion. In both cases, meaning arises from order. A small change in sequence can alter a protein, shift a trait, or even create a new species. Each base attaches to a sugar molecule called deoxyribose, which connects to a phosphate group, forming the nucleotide. These nucleotides link in chains, sugar to phosphate, to build the backbone of DNA. The bases extend inward like steps on a spiral staircase, forming pairs held together by hydrogen bonds, 2 between adenine and thymine, 3 between cytosine and guanine. This pattern maintains structural balance. The total width of the double helix never varies, even though the letters differ. Such precision allows the molecule to replicate without error, to unwind and rewind billions of times without losing its form. When scientists first realized that the language of life could be reduced to 4 repeating symbols, they also understood that this simplicity made evolution possible. If the code had been too complex, mutations might have been lethal or meaningless. But with 4 bases and 3 base codons, sets of 3 letters that represent amino acids, DNA could store vast instructions with redundancy and resilience. Each triplet, or codon, corresponds to one of 20 amino acids, the building blocks of proteins. 64 possible codons exist, more than enough to describe life's entire vocabulary. This overabundance is no accident. It allows tolerance for mistakes. If a single base changes, the meaning may still remain intact. Reflecting on this, one can sense both elegance and humility in the design. DNA does not shout its importance, it whispers through repetition. Every cell in every organism follows the same simple syntax. Yet from that sameness emerges diversity beyond measure. Forests, oceans, brains, and song. The molecule is not intelligent, yet it encodes the potential for intelligence. It does not think, yet it makes thought possible. This is the quiet poetry of chemistry. When you consider your own body, remember that all its complexity rests upon these 4 letters repeating in intricate order. Your eyes, your voice, even the shape of your hands are written in sequences of adenine, thymine, cytosine, and guanine. These letters have been copied and recopied for billions of years through countless ancestors without ever changing their fundamental rule, A with T, C with G. It is a partnership as old as life itself, precise, balanced, and endlessly creative. The alphabet of being remains the same, even as the story it tells keeps evolving. Every time a cell divides, it faces an astonishing challenge: to make an exact copy of its DNA. This is not a casual task. A single human cell contains about 3 billion base pairs, and every one of them must be duplicated accurately before division. The process, known as replication, is one of nature's most precise and efficient mechanisms, a molecular act of copying perfection. It takes place inside the nucleus, where enzymes work in silent coordination, ensuring that the thread of life remains unbroken. Replication begins when the DNA double helix unwinds. Specialized enzymes called helicases move along the molecule, separating its 2 strands by breaking the hydrogen bonds between bases. The point of separation forms a structure known as the replication fork, where each exposed strand serves as a template for building a new complementary strand. The elegance of this mechanism lies in its simplicity. Because adenine always pairs with thymine and cytosine with guanine, each strand automatically dictates its opposite. No supervisor is required. Chemistry itself enforces the rules. Once the strands are open, an enzyme named DNA polymerase attaches to each template and begins adding new nucleotides, one by one, following the base pairing logic. It works at remarkable speed, adding hundreds of bases per second. On one side of the fork, polymerase moves smoothly in the same direction as the unwinding helix, creating the leading strand. On the other side, however, the geometry is reversed. Because DNA strands run in opposite directions, polymerase must copy the lagging strand in small fragments called Okazaki fragments, later joined by another enzyme, DNA ligase, to form a continuous chain. Throughout this process, a set of molecular proofreaders ensures accuracy. DNA polymerase itself checks each base as it adds it, correcting mismatches instantly. Additional repair enzymes patrol the finished strands, detecting and fixing tiny errors. The resulting error rate is extraordinary, roughly 1 mistake per billion bases. To put that in perspective, if you were to type a book of 1,000,000 pages, you would make less than one typo. Life maintains its stability through this discipline of correction. Replication is not just a mechanical act, it is the foundation of inheritance. Each new cell receives an identical copy of the genetic blueprint, ensuring continuity across generations of cells and, ultimately, organisms. In the developing embryo, replication occurs on a massive scale, building tissues and organs from a single fertilized egg. In adulthood, it continues quietly, repairing wounds, renewing skin, and maintaining the immune system. Every heartbeat is accompanied by billions of microscopic acts of renewal, guided by the molecular scribes that never rest. And yet, perfection is not absolute. Occasionally, a mistake slips through, a single misplaced base, or an extra segment inserted by accident. Most of these changes are harmless, some are corrected later, and a few create new traits that may persist. These rare errors, once seen as flaws, are the raw material of evolution. Without them, species would remain frozen, unable to adapt. In this way, replication is both faithful and creative, precise enough to preserve life, flexible enough to allow change. When you picture replication, imagine two strands gently parting and reforming in quiet rhythm, like waves meeting and separating on a shore. The enzymes move with purpose, not haste, maintaining a balance between speed and accuracy that has kept life stable for billions of years. It is chemistry behaving as though it knows responsibility, a choreography written once and repeated perfectly ever since. Within each of your cells, this silent performance happens thousands of times a day, unseen yet vital. Through it, the story of life continues without pause, each line rewritten, each letter renewed. For all its precision, DNA replication is never completely flawless. In every living cell, tiny errors occasionally slip through the rigorous proofreading system. A misplaced base, a missing letter, or a small duplication. These are mutations, and although they might seem like imperfections, they are in fact the quiet architects of evolution. Without them, life would have remained unchanged from its earliest forms, unable to adapt or diversify. Mutation is nature's form of experimentation, a subtle test of possibility that continues in every organism, every moment. There are several kinds of mutation. A point mutation changes a single base pair, substituting one letter for another. If the altered codon still codes for the same amino acid, the change is silent. Sometimes, however, the result shifts a protein's shape or function. Insertions and deletions add or remove bases, potentially altering the reading frame of a gene, a single slip that can transform the entire message. Larger structural changes, such as duplications, inversions, or translocations, can rearrange entire sections of chromosomes. Most of these variations are corrected or rendered harmless, but a few provide new traits that natural selection can favor. The human body, like all living systems, contains countless safeguards to prevent harm. DNA repair enzymes continuously scan for mismatches and chemical damage. Ultraviolet radiation, reactive oxygen, and certain chemicals can break or alter DNA, but cells respond with restoration systems that patch and rebuild the affected segments. Still, absolute perfection would mean stasis. Mutation, even in its smallest form, ensures flexibility, an escape from uniformity, a way for life to explore its own potential. Consider how every species that has ever lived, from ancient trilobites to modern humans, emerged from a lineage shaped by error. Each accidental change that offered advantage, sharper vision, better metabolism, resistance to disease, became part of the ongoing story of evolution. Those that caused harm simply faded away. Through billions of years of trial and revision, the genome of every organism has been tuned by this patient process. Mutation, selection, inheritance, together they form the slow pulse of life's creativity. Even within the human body, mutation is both a risk and a necessity. In the immune system, for example, cells deliberately mutate their own DNA to create vast libraries of antibodies capable of recognizing countless pathogens. It is a controlled chaos, a biological improvisation that keeps the body safe. Elsewhere, mutations may lead to cancer or genetic disease, reminders that nature's experiments carry cost as well as progress. Yet, across the grand scale of time, it is variation, not perfection, that sustains existence. As you settle deeper into rest, let this balance bring a sense of calm. The same process that allows stars to burn and oceans to move also guides the quiet renewal inside your body. Each night, as your cells repair themselves, they follow patterns written in DNA, fixing what can be fixed and leaving behind the rare changes that may one day matter. Even in sleep, this invisible work continues, keeping you part of the living experiment that began billions of years ago. As you drift, remember that rest itself is a kind of repair, a pause that restores order while allowing gentle renewal. In the space between one heartbeat and the next, countless molecules align and mend, preserving the code that defines you. Mutation gives life room to change. Rest gives it time to recover. Both serve the same quiet purpose: continuity. And so, beneath your awareness, the script of life keeps writing, error by error, correction by correction, shaping the endless story of becoming. DNA may hold the instructions for life, but it cannot act alone. Its letters are static until read, its messages inert until expressed. The true power of the genetic code lies in its ability to guide the construction of proteins, the working molecules of the body. Proteins form skin and bone, build muscles and enzymes, and regulate every reaction that sustains life. The path from gene to protein is called gene expression, and it unfolds in two essential steps: transcription and translation. In transcription, the information stored in DNA is copied into a temporary molecule called messenger RNA, mRNA. Inside the nucleus, the enzyme RNA polymerase binds to a region known as a promoter, the starting signal of a gene. The DNA strands separate, and polymerase moves along one of them, reading its sequence and assembling a complementary RNA strand. This RNA copy mirrors the DNA's instructions, except that it uses uracil in place of thymine. Once complete, the mRNA detaches, carrying the genetic message safely out of the nucleus and into the cytoplasm, where protein synthesis begins. This process is remarkably selective. Not every gene is active in every cell. Liver cells, neurons, and muscle fibers all share the same DNA, yet each expresses a distinct set of genes suited to its purpose. This regulation comes from a network of chemical signals and proteins that switch genes on or off as needed. In this way, the genome acts like a vast library, the same collection of books available to all, but each cell reading only the volumes required for its role. Once outside the nucleus, the mRNA encounters the ribosome, a complex molecular machine built of proteins and ribosomal RNA. The ribosome reads the mRNA's sequence in triplets called codons, each representing one amino acid. Another molecule, transfer RNA, tRNA, carries amino acids to the ribosome, matching each codon with its corresponding building block. The ribosome links these amino acids together, forming a growing chain, a polypeptide, that will fold into a specific shape to become a functioning protein. The moment of folding is critical. A protein's shape determines its job. An enzyme's pocket fits the molecule it acts upon. A receptor's surface recognizes a hormone like a key meeting its lock. If even one amino acid is misplaced, the shape may change, and with it the protein's ability to function. Yet in most cases, the process unfolds flawlessly, guided by the physics of molecular interaction. Nature has rehearsed this choreography billions of times, refining it until each step occurs in silent precision. From a human perspective, this conversion of sequence to substance is breathtaking. A pattern of 4 chemical letters becomes an arm lifting, a thought forming, or a heartbeat pulsing. The code does not merely describe life, it performs it. Every movement, emotion, and breath begins with the quiet reading of genes. The language of DNA becomes the rhythm of metabolism, the structure of bones, the chemistry of memory. Reflecting on this flow, from gene to RNA to protein, reveals the continuity that runs through all living beings. The same molecular script that builds a fern's leaf also shapes a whale's fin and a human hand. Evolution did not rewrite the code, it simply rearranged the sentences. Within that shared grammar lies the unity of all organisms. If you listen to the body's internal stillness, this transformation is happening even now. Every cell, every moment, translates chemistry into existence. And though it occurs far beneath awareness, its effect is visible in everything you are, proof that the letters of life are never silent. Deep inside every living cell sits a structure no larger than a virus, yet more productive than any factory humankind has ever built. This is the ribosome, a molecular workshop where information becomes substance, where the language of nucleotides is translated into the tangible form of proteins. Without ribosomes, DNA would remain a silent archive, its messages unread. With them, the body comes alive, manufacturing enzymes, hormones, antibodies, and the countless structural proteins that give tissues their strength and shape. A ribosome is built from 2 subunits, large and small, each made of ribosomal RNA, rRNA, and proteins. These components assemble like interlocking gears, forming a machine with one purpose: to read the sequence of messenger RNA and build a corresponding chain of amino acids. Once an mRNA strand leaves the nucleus, it threads itself into the ribosome's entry channel. The ribosome locks onto it and begins to read, codon by codon, 3 letters at a time. Each codon corresponds to one of 20 possible amino acids, and each amino acid is delivered by a specialized carrier called transfer RNA. Each tRNA has a loop that matches the codon and a tail that holds its specific amino acid. When the tRNA's loop aligns perfectly with the mRNA codon, the ribosome catalyzes the bond between neighboring amino acids. The chain lengthens, curling outward like a growing thread. The process continues with remarkable efficiency, up to 20 amino acids added every second, all powered by molecules of ATP, the cell's universal energy currency. Once a stop codon is reached, the finished protein is released to fold into its functional shape. This precision assembly occurs billions of times per second across all living things. In bacteria, a single ribosome can complete a small protein in under a minute. In human cells, thousands of ribosomes work simultaneously, forming a polyribosome, each reading a different section of the same mRNA to multiply production. It is a scene of controlled chaos, microscopic workers moving in rhythm, guided by nothing more than chemical attraction and spatial geometry, yet achieving perfect order. After synthesis, the new polypeptide often travels to the endoplasmic reticulum, where it is folded and modified. Chaperone proteins assist in this process, ensuring that each molecule assumes its correct configuration. Misfolded proteins are recognized and dismantled, preventing malfunction. Some proteins remain in the cytoplasm, others are packaged into vesicles and sent to their destinations, to membranes, organelles, or secretory pathways. The ribosome, having completed one assignment, quickly binds another mRNA, ready to begin again. Reflecting on this process reveals the elegance of molecular logic. The ribosome is not alive in itself. It has no awareness, no decision-making, yet it embodies coordination and reliability beyond any human invention. It turns the abstract code of DNA into the physical essence of life, a transformation repeated trillions of times each day within your body. Every muscle contraction, every thought, every immune response depends on the silent labor of these molecular artisans. You might imagine, as you breathe slowly, millions of ribosomes in your cells working at once, tiny workshops powered by invisible hands, each following instructions written long before your birth. Their rhythm is steady, almost musical, a quiet hum beneath the awareness of the mind. It is a hum that began billions of years ago and has never ceased, echoing through the lineage of all living creatures. Within their tireless work lies the reason you exist, a translation, molecule by molecule, of life's original language. If you could uncoil all the DNA contained in a single human cell and lay it end to end, it would stretch for nearly 2 meters, yet it fits within a nucleus only a few millionths of a meter across. The secret to this impossible compression lies in organization. DNA is not stuffed into the cell like tangled thread. It is folded, looped, and wrapped with exquisite precision into structures that protect it while keeping it accessible. This packaging transforms a fragile molecule into a stable archive that can endure for decades, sometimes centuries, without losing its message. The fundamental unit of this organization is the nucleosome. In this structure, DNA winds almost 2 turns around a cluster of 8 proteins called histones. Each histone acts like a spool, and the DNA like a flexible ribbon wrapped around it. Roughly every 200 base pairs, the strand loops from one histone to the next, forming a chain that resembles beads on a string. These nucleosomes coil further into fibers that twist and fold, eventually condensing into the familiar chromosomes seen under a microscope during cell division. The result is not chaotic compression but an intricate hierarchy of loops and coils, each level reinforcing the balance between density and accessibility. Histones are not merely passive supports, they actively participate in gene regulation. Chemical groups, such as methyl or acetyl, can attach to histone tails, loosening or tightening the DNA's grip. When the grip relaxes, genes become available for transcription. When it tightens, those genes fall silent. This system, part of what scientists call epigenetic regulation, allows the same DNA to behave differently in different cells. A neuron and a skin cell contain identical genomes, but distinct chemical markings determine which parts are read and which remain closed. Beyond the nucleus's boundaries, the architecture continues. The DNA's organization ensures that replication, repair, and transcription occur efficiently, each process finding its place within the crowded space. Loops bring distant genes into proximity, enabling coordinated regulation. Specialized proteins anchor the DNA to the inner nuclear membrane, shaping territories where chromosomes occupy distinct regions. The nucleus, far from being an amorphous blob, is a living map of activity, every position purposeful. The sophistication of this design is staggering. It allows a cell to pack enormous information into minimal volume while retaining instant access to what it needs. The genome's folding is not static. It shifts continuously as genes turn on and off, as the cell divides, differentiates, and responds to signals. Even the act of reading DNA requires local unfolding, a delicate opening of space before the strands return to rest. The entire nucleus is a breathing structure, expanding, contracting, and rearranging itself in rhythm with the cell's life. When scientists first visualized this organization through electron microscopy and fluorescent tagging, they saw not a dense tangle but a landscape, valleys of inactive DNA and bright ridges of expression. The nucleus resembled a living city, its districts glowing and dimming as day passed into night within the molecular world. This constant motion embodies balance, order sustained by flexibility. You might picture, as you rest, your own cells holding these spirals within spirals, scrolls rolled carefully, then folded again, stored but never lost. Inside every nucleus, 2 meters of genetic text are kept safe without tearing or tangling, ready to be read at any moment. The perfection of that arrangement is silent and unseen, yet it works tirelessly, every instant of your life. It is the architecture of memory itself, proof that structure can be both protective and alive, that even in confinement, there is grace in order. Every living organism depends on one essential rhythm, the renewal of its cells. In the human body, roughly 2 trillion cells divide each day, replacing those that age or wear out. Behind this constant renewal lies one of nature's most reliable processes, cell division, the event that ensures DNA, once copied, is distributed evenly between new generations of cells. It is how a single fertilized egg becomes a body of trillions, and how tissues repair themselves throughout life. The dance of division is as precise as it is universal, balancing growth with continuity. There are 2 main forms of division: mitosis and meiosis. Mitosis occurs in ordinary cells, skin, muscle, bone, and countless others, to produce 2 genetically identical daughter cells. The process begins in interphase, when the cell grows and replicates its DNA, forming 2 identical copies of each chromosome called sister chromatids. Next comes prophase, where the chromosomes condense into visible threads and the nuclear envelope dissolves. Tiny structures known as spindle fibers form, reaching out like taut strings across the cell. During metaphase, the chromosomes line up neatly at the cell's equator, attached to these spindles by protein structures called kinetochores. In anaphase, the chromatids separate, pulled toward opposite poles with perfect symmetry. Finally, in telophase, new nuclear envelopes form, and the cell pinches in two, a process called cytokinesis, leaving behind 2 identical copies of the original. Each of these steps happens in microscopic silence, yet the precision rivals any human machine. Every chromosome must attach correctly before separation begins. Even a single mistake can lead to imbalance, causing genetic disorders or cell death. To prevent this, cells rely on molecular checkpoints that pause division until errors are corrected. This careful supervision ensures that each daughter cell inherits the complete and accurate blueprint of life. In the reproductive cells, sperm and egg, the story follows a different path. Meiosis reduces the number of chromosomes by half, producing gametes that, when united, restore the full complement. It unfolds in 2 rounds of division. In the first, homologous chromosomes, one from each parent, pair up and sometimes exchange segments, a process called crossing over that shuffles genes to create diversity. The second division separates the chromatids, yielding 4 unique cells, each containing a single copy of the genome. When 2 gametes meet at fertilization, their chromosomes combine to form a new individual, carrying traits from both ancestral lines. Thus, meiosis weaves genetic variability into the stability of inheritance, ensuring that each generation is both connected to and distinct from the last. This balance between accuracy and variation is life's greatest compromise. Perfect copying maintains function, but without recombination and mutation, there would be no evolution. Division is therefore not merely duplication, it is renewal with potential. Every living thing, from the smallest bacterium to the largest tree, obeys this rule. The continuity of life, through cycles of replication and exchange. You might imagine the cell as a clock that resets itself after every tick. The chromosomes align, separate, and return to stillness, waiting for the next signal. The rhythm is both mechanical and sacred, the heartbeat of existence at its smallest scale. Each new cell carries not only the complete record of its past, but also the capacity to adapt to the future. When you rest, your body continues this work without effort. Millions of cells divide in your skin, your bones, and your organs, each one a perfect continuation of you. Division is life's quiet assurance that nothing living truly stands still. It only transforms, one cell at a time. Every generation is a remix of what came before. During the formation of eggs and sperm, nature reshuffles the deck of heredity in a process both subtle and profound. This is crossing over, a molecular exchange that occurs during meiosis, when homologous chromosomes, one from each parent, align and trade segments of DNA. It is this exchange that gives rise to genetic diversity, ensuring that no 2 siblings, except identical twins, are ever exactly the same. In the quiet space of the cell, diversity is born not from chaos, but from carefully orchestrated recombination. At the start of meiosis, each chromosome duplicates itself into 2 identical sister chromatids. The matching pairs then come together, forming structures called bivalents, held in tight contact along their length. At certain points, the chromatids break and reconnect, swapping equal stretches of genetic material. These crossing points, called chiasmata, are where genes exchange positions between maternal and paternal DNA. When the process ends, each resulting gamete carries a unique combination of alleles, different versions of genes drawn from both family lines. The precision of this exchange is remarkable. Specialized enzymes, such as recombinases, guide the cutting and rejoining process, ensuring the segments match perfectly. If a crossover happens too often, it can disrupt chromosome integrity. If it happens too rarely, genetic uniformity might threaten a population's ability to adapt. Evolution has therefore tuned the frequency of crossing over to balance stability with novelty. On average, each pair of human chromosomes experiences about 1 or 2 crossovers during meiosis, creating nearly infinite possible combinations. The result is variation at the deepest molecular level, a foundation for evolution's creative power. It is through this quiet shuffling that populations remain resilient. Crossing over introduces combinations of genes that may yield new strengths, resistance to disease, improved metabolism, or subtler changes that affect behavior and form. These differences become the raw material upon which natural selection acts. A species unable to generate variety would struggle to survive environmental change. Thus, the act of exchange within each cell mirrors a much larger truth. Adaptability depends on connection. When scientists first observed chiasmata under early microscopes, they saw the literal points of intersection between parental legacies, a physical record of unity. Later, Genetic mapping revealed that the distance between genes could be estimated by how often they crossed over. This insight allowed researchers to create linkage maps, charting genes long before DNA sequencing existed. The closer two genes were on a chromosome, the less likely they were to separate, bound by the same stretch of inheritance. It was as if nature herself kept a ledger of probabilities inside the nucleus. Reflecting on this process invites a sense of quiet wonder. Crossing over is not loud or visible. It happens silently, deep within the germ cells, long before birth. Yet from this silent act comes every face, every pattern of freckles, every shade of hair and eye. It is the biological counterpart of creativity, generating novelty within boundaries, improvisation guided by rules. If you imagine it now, think of two ribbons gently intertwined, trading threads before parting ways. The exchange leaves each one altered but still whole, enriched by contact. That is the essence of crossing over, unity leading to individuality. And through countless repetitions of this dance, life ensures that its next verse will never sound exactly like the last. Over billions of years, the 4 letters of DNA have written every life form that ever existed, revising, reordering, and refining themselves through time. Evolution is the grand editor of this molecular text, turning small genetic changes into vast biological variety. It works without design or destination, guided only by selection and chance. From bacteria adapting to new nutrients to birds evolving flight, the same invisible script continues to rewrite itself, one letter, one mutation at a time. The modern understanding of evolution began when Charles Darwin described natural selection in the 19th century. He proposed that small variations arise naturally within populations, and those best suited to survive in their environment tend to reproduce more successfully. What Darwin could not yet know was the source of those variations. A century later, molecular biology revealed that DNA is that source. Mutations, recombinations, and copying errors provide the raw material, while the environment acts as the editor, preserving what works and discarding what does not. This union of genetics with Darwin's idea became known as the modern synthesis, a framework that explained how microscopic changes could produce macroscopic transformation. Every species, including humans, carries in its genome a record of its journey through time. Fossils preserve bones and shells, but DNA preserves instructions, a living archive of evolutionary history. Within your own cells are ancient sequences inherited from creatures that lived long before humans appeared. Some genes are nearly universal. The same code that builds a human eye also helps a jellyfish sense light. Such conservation shows how evolution favors efficiency, reusing effective designs rather than reinventing them. The difference between species lies not in entirely new inventions, but in how old ones are modified and expressed. Evolution works slowly, but it never stops. It operates in every generation, every replication. A bacterium that develops resistance to an antibiotic does so because one tiny mutation, once neutral, suddenly provides an advantage. The same logic applies to the immune system refining its defenses or to crops adapting to new climates. The mechanism is simple, but its consequences are infinite. The genome is both a memory and a workshop, carrying the traces of its past while experimenting with its own future. In the human genome, for example, over half of the DNA consists of remnants of ancient viruses and repeated sequences that once moved freely within chromosomes. Some of these fragments have become useful, helping regulate gene expression or form new genetic structures. Evolution wastes nothing. It transforms debris into function. Every species is thus both an outcome and an ongoing experiment. The tempo of change varies. In some organisms, evolution proceeds gradually. In others, sudden environmental shifts accelerate it. Extinction and innovation are partners in this rhythm. As some genes vanish, others emerge. Over long timescales, the cumulative effect reshapes ecosystems, generating new forms from old templates. The same four bases remain, but their order, spacing, and control evolve endlessly. DNA in this way behaves like language. The alphabet stays constant, but the stories never repeat. As you rest, it is humbling to realize that evolution continues even now, in every cell division, in every living generation. The molecule inside you has been refined through unbroken ancestry, reaching back to the first self-replicating structures in Earth's primordial seas. You are not separate from that story. You are one of its latest sentences. As you drift into sleep, imagine the code still quietly at work, copying, adjusting, preserving. Evolution does not sleep, but you can. While you rest, the same molecular forces that shaped galaxies of life carry on their patient work within you. Every breath and heartbeat belongs to an ancient rhythm of adaptation and renewal. In the stillness of sleep, the story of change continues, letter by letter, keeping life forever in motion. Every living organism is a mosaic of the past. Within each strand of DNA lie traces of earlier forms, silent witnesses to the long ancestry of life. Some genes remain almost unchanged across species and epochs, preserved because they perform roles too essential to be replaced. Others have drifted, duplicated, or mutated, leaving faint echoes of vanished creatures. When scientists sequence DNA today, they are reading not just the instructions for a single organism, but the molecular history of an entire planet. One of the most striking examples of this continuity is found in cytochrome C, a small protein involved in cellular respiration. Its sequence of amino acids is nearly identical in humans, fruit flies, and yeast. Such conservation tells a story. Once evolution found a design that worked, it held on to it. Changing even one part of this molecule would risk breaking the chain of reactions that sustain life. These ancient proteins act as molecular fossils, preserving the shape and chemistry of the earliest cells that breathed oxygen more than 2 billion years ago. Other relics of the past appear in the form of pseudogenes, fragments of once functional genes that have lost their ability to code for proteins. They remain in our genome like faded pages from an old manuscript, copies of instructions no longer used. Some pseudogenes record evolutionary experiments that failed. Others have acquired new regulatory roles, influencing which genes switch on and off. The genome, far from being a neat library, is a palimpsest, a layered text, rewritten over time but never entirely erased. Among the most intriguing molecular fossils are endogenous retroviruses, remnants of viral infections that invaded the germline of our ancestors. When certain viruses infect a cell, they insert their own genetic material into the host's DNA. If that cell happens to be reproductive, the viral sequence becomes part of the lineage, passed on like any other gene. Over millions of years, these insertions accumulated, gradually losing their infectious potential, but remaining as part of our genome. In humans, they make up roughly 8% of our DNA. Some of these ancient viral genes have even been repurposed. One helps form the placenta, a structure that makes mammalian reproduction possible. In this way, an ancient infection became essential for life. Through comparative genomics, scientists can now trace how genes travel and transform across species. They build evolutionary trees not from fossils in rock, but from mutations in sequence. A single base change that appears in both humans and chimpanzees, but not in gorillas, marks a common ancestor about 6 million years ago. In this way, DNA becomes a timeline written in molecular script, each letter a clue to origin. Reflecting on this, one can sense that the genome is not simply a manual for building bodies, but a diary of survival. Each sequence represents something that once worked well enough to endure. The arrangement of bases in your cells carries whispers of ancient oceans, microbial mats, and early vertebrates. You are, at the most literal level, composed of the past. If you imagine this history, see your DNA as a long scroll filled with overlapping handwriting, new lines inscribed atop older ones, traces of ink showing through. The present life you live is written across pages that began billions of years ago. Every heartbeat continues a line that started with a molecule floating in the sea, learning to copy itself. The continuity between you and those first cells is unbroken. Inside every nucleus, the story of life remains whole, ancient, silent, and still being written. At the dawn of the 21st century, humanity accomplished one of its most ambitious scientific goals, the complete mapping of its own genetic code. The Human Genome Project sought to identify every gene in human DNA and determine the precise order of its 3 billion base pairs. It was a collaboration that united laboratories across continents, involving thousands of scientists, computers, and instruments working in quiet synchrony. The result was more than a list of letters. It was a mirror held up to life itself, revealing the full scope of what it means to be human at the molecular level. The project officially began in the late 1980s, inspired by rapid advances in sequencing technology. For centuries, biology had relied on observation. Now, it could turn to direct reading. Researchers broke the genome into fragments, sequenced each piece, and used computational algorithms to reassemble the data like a vast puzzle. The task required new methods of automation and data management, the foundations of modern bioinformatics. By the year 2003, the first draft of the human genome was declared complete. It showed that our DNA contains about 20,000 protein-coding genes, far fewer than many had expected. The complexity of humans, it turned out, arises not from the number of genes, but from how they are regulated, combined, and expressed. The publication of the genome was a moment of collective astonishment. For the first time, people could look at the letters that underlie every cell in their bodies. It was as though the species had turned its gaze inward, reading the text that evolution had been writing for billions of years. Scientists found long stretches of non-coding DNA, once dismissed as junk, later discovered to play crucial roles in controlling when and where genes are activated. They uncovered patterns of repeats, silent sequences, and ancient viral insertions, the molecular archaeology of our species. The project's legacy reaches far beyond knowledge for its own sake. It revolutionized medicine, giving rise to genomic medicine, where treatments can be tailored to a patient's genetic profile. Disorders, once mysterious, can now be traced to single mutations. Cancer, once described by its location, can be characterized by its molecular signature, leading to targeted therapies. Genetic testing has expanded preventive care, allowing families to understand inherited risks long before symptoms appear. What once took years of laboratory work can now be accomplished in hours by automated sequencers smaller than a briefcase. Ethical questions emerged alongside these discoveries. Who owns genetic information? How should privacy be protected when a single genome reveals so much about family members as well? These questions reminded the scientific community that knowledge carries responsibility. To address such concerns, the Human Genome Project included a dedicated ethics and policy division, ensuring that the science of reading life's code was matched by the wisdom to use it carefully. Reflecting on this milestone reveals a deep shift in how humanity perceives itself. For the first time, we could see the unity beneath our differences. Across continents and cultures, every human genome is more than 99% identical. That shared sequence underscores what philosophy had long claimed but never proven with such clarity, that our diversity is written upon a foundation of profound similarity. If you imagine the genome as a library, the Human Genome Project opened its doors for the first time, illuminating shelves that had been locked since the dawn of life. And while the reading has only begun, the light it shed continues to guide exploration, a calm, persistent glow inside the vast darkness of the unknown. Every cell in the human body carries the same DNA, yet no two cells behave exactly alike. A neuron transmits electrical impulses, a liver cell breaks down toxins, a muscle fiber contracts. The difference is not in the genetic script itself, but in how it is read. Genes do not operate in isolation. They exist within a living context, constantly responding to signals from the environment, diet, stress, temperature, and experience. This dialogue between genes and surroundings defines much of what we are. The genome provides the blueprint, but the world provides the conditions that decide which parts of that plan unfold. This interaction begins even before birth. Chemical cues in the womb influence which genes activate as an embryo develops. After birth, exposure to light, nutrients, and social contact continues to shape gene expression. The process that mediates this flexibility is known as epigenetics, the study of how chemical modifications on DNA and its associated proteins control whether genes are turned on or off. The sequence itself remains unchanged, but its accessibility shifts like the pages of a book being opened or closed. These changes can last for days, years, or even lifetimes, affecting metabolism, immunity, and behavior. One of the best-known epigenetic mechanisms involves methylation, in which small methyl groups attach to certain cytosine bases in DNA. When added to a gene's promoter region, these marks act like punctuation that silences transcription. Another mechanism, histone modification, alters how tightly DNA wraps around histone proteins. Acetylation tends to loosen the wrapping, encouraging gene activity. Deacetylation tightens it, reducing access. Together, these molecular switches fine-tune the flow of information, adapting the cell's behavior to its circumstances. External factors constantly influence these epigenetic marks. Nutrition, for instance, affects methylation patterns. A diet rich in folate or vitamin B12 provides the molecules needed to add methyl groups, while famine can erase them. Stress, too, can reshape the epigenetic landscape. Studies in both humans and animals show that prolonged stress can suppress genes involved in growth and immunity, while enhancing those that prepare the body for vigilance. In this way, experience writes itself into biology, leaving a signature that persists long after the event has passed. Even learning and memory involve epigenetic regulation. When neurons form new connections, certain genes must activate temporarily, then quiet again. These bursts of expression are guided by small molecular markers that tell the genome when to speak and when to rest. The brain, therefore, does not only store information in networks of cells, it also marks the very genes that enable learning. Your memories, in a sense, are supported by the chemistry of adaptation. Perhaps most striking, some epigenetic changes can be inherited. If a parent experiences famine, trauma, or environmental toxins, those exposures can sometimes influence gene regulation in their offspring. The underlying DNA sequence remains identical, but its expression shifts, preparing the next generation for conditions similar to those the parents faced. While this inheritance is not absolute, it demonstrates that life records more than mutations alone. It also remembers conditions and context. Reflecting on this reveals how dynamic the genome truly is. DNA is not a rigid instruction manual, but a living document that the environment edits constantly. In every moment, your body is negotiating between stability and flexibility, preserving the essence of its design while adapting to experience. You might imagine your genes as instruments and the environment as the musician. The notes remain the same, but each performance sounds different depending on touch, timing, and emotion. Within that interplay lies individuality, the music of life composed anew in every cell, every day. Few natural experiments reveal the relationship between genes and environment as clearly as identical twins. They begin with precisely the same DNA sequence, the result of a single fertilized egg dividing into two embryos. For a moment in time, they are molecular copies, same genes, same instructions. Yet as they grow, subtle differences begin to appear. One may develop a different temperament, another a distinct health condition, or even unique preferences and talents. These divergences show that the genome sets the foundation, but life's experiences shape the outcome. Identical twins are living proof that biology and environment are inseparable partners in the making of individuality. Early twin studies, conducted in the 20th century, sought to measure heritability, the proportion of variation in traits that can be attributed to genetics rather than environment. Researchers compared twins raised together and apart, observing patterns in intelligence, personality, and susceptibility to disease. Their findings revealed that many human characteristics have a strong genetic component, yet none are entirely predetermined. Even when DNA is identical, the expression of that DNA diverges over time. Modern genetics has shown why. Although identical twins start with the same genome, their epigenetic patterns begin to differ as they experience distinct environments, diets, stresses, and lifestyles. Chemical marks such as DNA methylation accumulate differently in each twin, turning certain genes on or off. Over decades, these marks reshape cellular behavior, influencing metabolism, immune response, and even mental health. If one twin smokes, exercises more, or encounters more stress, those differences leave molecular traces visible to scientists studying their cells. The longer twins live, the more distinct these marks become, showing that identity extends far beyond sequence. The body's tissues also evolve independently. Somatic mutations, small changes that occur as cells divide, can accumulate differently between twins, especially in rapidly renewing tissues such as skin or blood. While most of these mutations are harmless, they contribute to biological individuality. Thus, even at the microscopic level, two genetically identical people gradually diverge into unique molecular landscapes. This divergence explains why one twin might develop a disease while the other remains healthy. Conditions like diabetes, depression, or cancer often arise from a complex interplay between genetic predisposition and environmental triggers. The DNA provides a possibility, not a destiny. A gene variant might increase risk, but diet, exposure, and stress determine whether that risk becomes reality. Understanding this interplay has shaped personalized medicine, reminding us that genetic similarity does not guarantee identical outcomes. Beyond biology, the story of twins also reflects the subtler layers of individuality, memory, emotion, and choice. Shared DNA cannot account for how two minds interpret the same experiences. The human brain, shaped by both chemistry and circumstance, grows through feedback from the world. Each interaction, a friendship, a challenge, a moment of joy, leaves marks that no molecule can predict in advance. The twins' shared origin becomes two separate lives, each a different response to the same starting code. Reflecting on this, it becomes clear that identity is both inherited and created. DNA provides the structure, living fills in the meaning. Even the smallest variation in experience alters how genes perform their tasks, weaving personality and fate from a single source. If you think of identical twins as two mirrors facing one another, what stands between them is not opposition but time. Each reflection gathers its own dust, light, and color. The genome began the same, but the world painted differently upon each canvas. In this way, life proves that sameness and difference can coexist, written in the same four letters yet composed into infinitely varied stories. Every experience leaves a mark, not just on the mind, but on the molecules that sustain life. Within each cell, the genome is constantly being annotated by small chemical notes, additions that record what the cell has been through and how it should respond next time. This phenomenon, known as epigenetic memory, allows life to adapt without altering its DNA sequence. It is the reason identical cells can behave differently, and why events from the past can echo in generations yet to come. Epigenetic memory begins with the same mechanisms that regulate genes: methylation, histone modification, and chromatin remodeling. But instead of temporary adjustments, some of these marks persist. When a cell divides, the enzymes that copy DNA also replicate many of these chemical signals, ensuring that daughter cells inherit not only the genetic text but also the instructions for how to read it. In this way, a liver cell remembers that it is a liver cell, a neuron remembers its identity, and the body maintains its organization through countless divisions. Beyond development, this molecular memory can also reflect lived experience. Nutrient availability, stress, toxins, and even patterns of social interaction can alter the epigenetic landscape, leaving stable imprints on specific genes. Studies of individuals exposed to famine or severe trauma have revealed such marks decades later. In some cases, these modifications appear in their children as well, shaping metabolism, stress responses, or susceptibility to illness. Life, it seems, records more than sequence. It remembers circumstances. Not all memories are burdens. Epigenetic adaptation also allows resilience. Plants exposed to drought may transmit tolerance to their offspring. Immune cells that encounter pathogens keep a molecular record to respond faster next time. In humans, exercise, balanced nutrition, and emotional well-being can produce beneficial marks, improving cellular function. The genome, though silent, listens to the rhythm of daily life and adjusts accordingly. Within the brain, epigenetic memory takes on a more literal meaning. Learning involves waves of gene activation and silencing as neurons form new connections. These changes stabilize through chemical marks that keep certain genes available and others restrained. Thus, remembering a song, a face, or a lesson involves not only the firing of synapses but also tiny adjustments to the chromatin within those neurons. Each recollection is, in part, a molecular pattern, the physical trace of thought. The persistence of these marks blurs the boundary between biology and experience. It shows that identity is not fixed but responsive, that life's instructions include the capacity to learn and adapt. Our DNA, long viewed as static, is in truth dynamic, alive to its environment, carrying whispers of what has been encountered before. As you begin to drift towards sleep, remember that this same process continues inside you. During rest, cells repair DNA, replace worn molecules, and sometimes reset epigenetic marks that have drifted during the day. Sleep itself is a kind of renewal, a nightly act of editing in which the body chooses what to keep and what to release. If you imagine it, picture your cells as quiet libraries at night. The scribes move softly among the shelves, dusting pages, adding notes, or gently erasing what no longer serves. No effort is required on your part. This work happens in silence. As you breathe, the body restores balance, preserving the knowledge it needs for tomorrow. Epigenetic memory ensures continuity. Sleep ensures peace. Together they remind us that rest is not stillness but gentle preservation, the steady renewal of life's enduring code. DNA is an extraordinary molecule, durable enough to preserve life's code for billions of years, yet delicate enough to be damaged by a single stray photon of light or a reactive molecule of oxygen. Every cell in the body faces this vulnerability. Each day, within a single person, tens of thousands of DNA lesions occur, breaks, mismatches, or chemical alterations. Without constant repair, the genome would quickly accumulate errors too severe for survival. Life endures not because damage never happens, but because its repair systems work tirelessly, restoring order from disruption every moment. There are several distinct DNA repair pathways, each specialized for different types of harm. The simplest form of damage, the loss or modification of a single base, is corrected through base excision repair. Enzymes known as DNA glycosylases patrol the double helix, recognizing and removing damaged bases. They leave behind a tiny gap that is filled and sealed by other enzymes, DNA polymerase and DNA ligase, returning the sequence to its original state. For larger, more complex lesions, such as those caused by ultraviolet light, cells use nucleotide excision repair, a system that cuts out an entire section of the strand surrounding the defect and replaces it with a fresh copy. Mistakes that slip through replication are caught by the mismatch repair mechanism. This pathway identifies incorrect base pairings, such as an adenine paired with a cytosine, and restores the correct match. Meanwhile, double-strand breaks, among the most serious injuries a chromosome can suffer, trigger repair through either homologous recombination, which uses an undamaged sister chromatid as a guide, or non-homologous end joining, which directly reconnects the broken ends. Each system acts like a specialized team, responding to specific emergencies in the molecular landscape. The coordination of these mechanisms is remarkable. As soon as damage occurs, sensors detect distortions in the DNA's shape and summon the appropriate enzymes. Signaling cascades ensure that the cell pauses division until repairs are complete. If the harm is beyond repair, the cell may self-destruct through apoptosis, protecting the organism by sacrificing itself. This discipline ensures that errors do not multiply and that only stable DNA is passed on to daughter cells. Some organisms have developed extraordinary repair capabilities. The bacterium Deinococcus radiodurans can survive radiation doses thousands of times higher than those that would kill a human, reassembling shattered DNA within hours. Even in humans, cells demonstrate resilience. The nervous system, skin, and immune cells constantly mend their DNA under the influence of sunlight, toxins, and oxidative stress, maintaining fidelity despite relentless exposure. Over time, however, small scars remain, single bases that escape correction, fragments lost, telomeres gradually shortening with each replication. Aging itself can be seen as the accumulation of such molecular wear, yet even here, the repair process offers hope. Lifestyle choices such as balanced diet, exercise, and restorative sleep enhance the cell's ability to repair damage, largely by reducing oxidative stress and supporting the enzymes that rebuild the genome. Reflecting on this system evokes quiet admiration. Life does not depend on avoiding harm but on recovering from it. Perfection is not its goal, persistence is. Every cell's survival depends on the patient repetition of repair, day and night, without recognition or pause. If you picture it, imagine your DNA as a long, delicate ribbon, its surface continuously inspected by invisible caretakers. When a break appears, they mend it. When a letter fades, they restore it. Their work is unending, yet they never tire. The body's endurance comes from this quiet diligence, resilience built not from strength alone but from care. The same patience that keeps molecules whole also keeps life continuous, one repair at a time. Among the countless influences that have shaped DNA, few are as surprising or as creative as viruses. To most minds, they represent harm, agents of infection and disease. Yet, on the molecular level, viruses are also collaborators, contributors to evolution's toolbox. Over billions of years, they have exchanged genetic material with their hosts, leaving behind fragments that have become permanent residents of the genome. Life, in its long history, has borrowed freely from what once threatened it. A virus is little more than a shell of protein enclosing a short piece of nucleic acid, DNA or RNA. Lacking the machinery to reproduce on its own, it must invade a host cell and use that cell's systems to make copies of itself. When a retrovirus infects, it converts its RNA into DNA using a special enzyme called reverse transcriptase. This viral DNA then inserts itself into the host genome, hijacking the cell's transcription machinery. Most infections are temporary, but sometimes the viral DNA remains, passed on each time the cell divides. If such an event occurs in an egg or sperm cell, the viral sequence becomes hereditary, an endogenous retrovirus embedded within the species' genetic fabric. In humans, these viral remnants make up an astonishing portion of our genome, roughly 8% of our DNA. Most are broken or inactive, molecular fossils of ancient epidemics. Yet some have been domesticated by evolution, repurposed to serve the host. One viral gene, syncytin, plays a crucial role in forming the placenta by helping cells fuse to create the barrier between mother and fetus. Without it, mammalian pregnancy would not exist in its current form. What began as infection became cooperation, an elegant example of nature's capacity to recycle adversity into advantage. Other viral elements act as regulators, controlling nearby genes. They can enhance expression in one context, suppress it in another, or even serve as switch points during embryonic development. In this sense, the genome is less a static archive and more a patchwork of genetic influences, some native, some adopted. The relationship between host and virus resembles a long conversation, alternating between conflict and collaboration. Over evolutionary time, both sides have adapted, refining their interactions into the delicate balance we observe today. This dynamic exchange continues. Many viruses that infect humans and animals still integrate small fragments of their genetic material into host cells. In some cases, these insertions contribute to genetic diversity. In others, they cause harm. Modern biotechnology, inspired by these natural mechanisms, now uses engineered viruses to deliver therapeutic genes, correcting mutations that cause disease. The very process once feared as invasion is now harnessed as healing. Reflecting on this, one can see that the boundary between self and other in biology is less distinct than it seems. The genome is not a fortress, it is a living document edited by the world around it. Viruses, once seen purely as enemies, are also teachers, showing how exchange and adaptation can drive creativity in evolution. Their role reminds us that life advances not only through defense but also through incorporation, by transforming the foreign into the familiar. If you picture your genome, imagine it not as a clean manuscript, but as a palimpsest filled with marginal notes from millions of encounters. Some are warnings, others additions that enrich the story. The viral passages that remain within you are silent now, yet they whisper of an ancient dialogue between life and its imitators. Out of that conversation emerged new possibilities, complexity, resilience, and the ability to change. In this way, even a virus, a particle without thought or purpose, became part of the story of becoming. In the vast history of biology, few discoveries have given humanity such direct influence over evolution as CRISPR. What began as an obscure bacterial defense system has become one of the most powerful tools ever devised, a way to edit genes with precision, speed, and accuracy. For the first time, life can consciously rewrite its own code. The letters that once took millions of years to change by chance can now be adjusted in hours within a laboratory. It is both a triumph of understanding and a test of responsibility. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, was first observed in bacteria in the 1980s. Scientists noticed unusual patterns of repeated DNA sequences, separated by unique spacers. For years, their purpose remained unknown. Then, in the early 2000s, researchers discovered that those spacers matched sequences from viruses that had previously infected the bacteria. CRISPR, it turned out, was a molecular memory bank, a record of viral invaders. When the same virus attacked again, the bacterium used a protein complex called Cas, CRISPR-associated, to recognize and cut the foreign DNA, neutralizing the threat. It was a natural immune system at the genetic level. The brilliance of CRISPR as a tool for gene editing lies in its simplicity. Scientists realized that by programming a short RNA molecule, called guide RNA, to match any chosen DNA sequence, the Cas9 enzyme could be directed to cut precisely at that location in the genome. Once the DNA is cut, the cell's own repair mechanisms take over, either joining the broken ends or inserting a new piece of DNA provided by researchers. This process allows the addition, deletion, or replacement of genes with remarkable accuracy. What once required years of selective breeding or random mutation can now be done in a single generation. The applications are immense. In medicine, CRISPR has already been used experimentally to correct genetic defects responsible for disorders like sickle cell disease, muscular dystrophy, and certain types of blindness. In agriculture, crops are being edited to resist pests, tolerate drought, or yield more nutritious fruit. Scientists are exploring ways to modify mosquitoes to stop the spread of malaria, or to engineer microbes that can clean pollution and capture carbon dioxide. Every field of biology has been touched by this single innovation. Yet CRISPR's power brings new ethical questions. The same technology that can cure disease could, in principle, be used to enhance traits, to select for intelligence, appearance, or strength. The line between therapy and design becomes thin. Some fear that altering the germline, the DNA passed to future generations, could have consequences we cannot yet predict. The simplicity of the tool makes it widely accessible, raising questions about regulation, misuse, and inequality. Humanity now holds the ability to shape its descendants in ways evolution never could, and that power demands restraint as much as imagination. Beyond its implications for people, CRISPR has also deepened our understanding of life's unity. A mechanism discovered in humble bacteria now enables medical breakthroughs in humans, showing once again that all living systems share common logic. What protects a microbe can guide a surgeon's hand. If you imagine it, picture the genome as a vast book written without spaces or punctuation, billions of letters long. For most of history, humans could only read it, never edit. CRISPR is the pen placed gently into our hands. Used wisely, it can correct the typos of nature, the mutations that cause suffering. Used carelessly, it could rewrite the meaning of life itself. The technology's true value will depend not only on precision but on wisdom, on our ability to see that power and humility must evolve together. The ability to rewrite the genetic code marks a turning point not only in science but in philosophy. For the first time, humanity holds direct authorship over the instructions that shape life itself. What was once the slow, patient work of evolution, guided by chance and filtered through natural selection, can now be altered with deliberate intent. This power invites both promise and peril. It compels us to ask, not just what can we do, but what should we do? The ethics of gene editing reach far beyond the laboratory, touching questions of identity, justice, and the meaning of being human. When scientists first demonstrated CRISPR's ability to modify embryos, the world's reaction was mixed awe and unease. On one hand, the potential to eliminate genetic diseases seemed like a moral imperative, to end inherited suffering, to give children freedom from disorders encoded at conception. On the other, the prospect of altering human heredity raised fears of unintended consequences. A single misplaced edit might affect not only one person but countless descendants. Once a genetic change enters the germline, it becomes part of humanity's shared legacy, irreversible and self-perpetuating. The stakes could not be higher. Ethical discussion often begins with somatic versus germline editing. Somatic edits, made to body cells, affect only the treated individual and can cure illnesses like sickle cell anemia or cystic fibrosis. These changes die with the patient. Germline edits, by contrast, modify eggs, sperm, or embryos and are passed to future generations. It is the latter that poses the deepest moral dilemma. Supporters argue that preventing genetic disease before birth spares future suffering. Critics respond that even small errors or unforeseen interactions could introduce new problems far worse than those solved. Biology is too complex to predict every ripple of change. Beyond safety, the issue of equity looms large. Advanced treatments often begin as costly technologies. If gene editing becomes a privilege of the wealthy, society risks a new form of inequality, not of opportunity or wealth, but of biology itself. Imagine a world where some can afford enhanced immunity, intelligence, or longevity, while others cannot. The ancient idea of equality before nature would dissolve. Ethical frameworks must therefore guard against a genetic divide, ensuring that medical benefits are shared fairly and used for healing, not hierarchy. There is also the concern of consent. An edited embryo cannot agree to the changes made in its genome. Future generations cannot approve the decisions that shape their biology. This absence of consent challenges our traditional notions of autonomy. It asks us to extend ethical consideration to those who do not yet exist, to act as custodians rather than controllers. Responsible science demands patience, to proceed only when safety and necessity are certain, and never for reasons of vanity or preference. Religious and philosophical traditions add further complexity. Some see gene editing as interference with natural order, an act of hubris that risks unbalancing the harmony of creation. Others view it as a continuation of humanity's role as caretaker, using knowledge to alleviate suffering and improve life. Both views recognize the profound responsibility that accompanies understanding. The question is not whether we will shape life, but how we will choose to shape it. History offers cautionary tales. In the 20th century, the misuse of genetics under the banner of eugenics led to discrimination and atrocities justified by pseudoscience. The lesson is clear. Knowledge without humility can corrupt. The new century's challenge is to ensure that the power to edit genes does not repeat old mistakes under a modern guise. Science must be guided by ethics, not ambition alone. International organizations and governments are now drafting frameworks to govern human gene editing. Many countries have banned germline modification until its consequences are better understood. Others allow limited research under strict oversight. This global dialogue between scientists, ethicists, policymakers, and the public reflects an awareness that the genome is not private property, but a shared inheritance of humanity. At its core, the ethics of editing demand balance. To deny all progress out of fear would be to turn away from compassion. To pursue it recklessly would be to invite harm. True wisdom lies in moderation, to recognize that power is not freedom but responsibility. If you picture the genome as a library, editing it is not vandalism nor blind worship of the text. It is stewardship. Every change should be made with reverence for what is written and humility about what is not yet understood. As the instruments of science grow sharper, conscience must grow stronger. The goal is not to perfect humanity, but to protect it, to ensure that, in mastering DNA, we do not forget the quiet laws of kindness and restraint that make us human in the first place. The double helix, once a symbol of biological discovery, has also become a cornerstone of modern justice. The same molecule that records ancestry and encodes life's blueprint now serves as one of the most powerful tools in criminal investigation. Forensic DNA analysis has reshaped how societies define truth, guilt, and innocence. It connects the smallest trace, a drop of blood, a single hair, a fragment of skin, to the identity of a person with extraordinary precision. Yet, as with every application of genetic knowledge, it brings both clarity and ethical complexity. Before the rise of DNA forensics, investigators relied on fingerprints, eyewitnesses, and circumstantial evidence. These methods, while useful, were prone to error and interpretation. The arrival of DNA profiling in the late 1980s transformed the field. The breakthrough came from the work of British geneticist Sir Alec Jeffreys, who discovered that specific regions of DNA vary greatly from person to person. By examining these differences, now known as short tandem repeats, STRs, scientists could create a unique genetic fingerprint for each individual. No two people, except identical twins, share the same pattern. Suddenly, identity could be measured at the molecular level. The process begins when forensic scientists collect biological evidence from a crime scene: blood, saliva, hair, or any tissue containing cells. DNA is extracted and purified, then amplified through a method called polymerase chain reaction, PCR, which creates millions of copies of targeted DNA fragments. Those fragments are then analyzed to reveal a pattern of STR markers. When compared with a suspect's sample or a genetic database, a match can confirm presence with an accuracy exceeding 1 in several trillion. Even degraded or tiny samples can yield results, allowing cases long considered unsolvable to find resolution. The impact of DNA evidence has been profound. It has exonerated the innocent and convicted the guilty, sometimes decades after a crime. The Innocence Project, founded in the United States, has used DNA testing to overturn hundreds of wrongful convictions, revealing the fallibility of earlier methods. Across the world, cold cases have been revived by a few preserved cells on clothing or stored slides. In these stories, the genome becomes both witness and record keeper, a silent observer preserving truth long after human memory fades. Yet with such power come challenges. Contamination, mishandling, or misinterpretation of evidence can still lead to error. The science is precise. Human application is not always so. There are also questions of privacy and consent. National DNA databases store profiles of millions of citizens, some convicted of crimes, others merely arrested or related to suspects. While these repositories aid investigation, they also raise fears of surveillance, misuse, or bias. DNA, unlike a fingerprint, reveals more than identity. It carries information about ancestry, health, and family ties. Protecting that information from abuse has become a growing concern. New technologies have expanded the reach of forensic genetics even further. Familial searching can identify potential relatives of a suspect when no direct match exists. Genetic genealogy, which combines public ancestry databases with crime scene DNA, has solved cases once thought impossible, including those dormant for decades. But such methods also blur boundaries between voluntary participation and public exposure. When one person uploads their genetic data to a genealogy site, they may inadvertently make much of their extended family traceable to investigators. This intersection between personal freedom and public safety is now one of the field's most debated issues. Beyond law enforcement, forensic DNA has applications in humanitarian work, identifying victims of natural disasters, wars, and human rights abuses. It reunites families separated by conflict and gives names to the lost, restoring dignity where history once left silence. In these cases, DNA becomes an instrument not of accusation but of compassion, connecting the living with their past. Reflecting on this duality, one sees how the genome bridges science and morality. It carries the power to reveal truth, but truth itself is neutral. It gains meaning only through how it is used. The same sequence that convicts can also forgive, that divides can also unite. The molecule has no bias. Interpretation gives it consequence. If you picture a courtroom, imagine a thread of DNA stretched between microscope and gavel, science and law joined by a single strand. That thread demands both accuracy and humility. Forensic genetics teaches that knowledge can serve justice only when handled with care, respect, and transparency. The code of life has entered the realm of law, and with it, the quiet reminder that truth, however powerful, must always serve humanity, never overshadow it. Every strand of DNA is a map, carrying coordinates not of places but of origins. Within the spiraled molecule of each human being lies a record of ancestry, migration, and adaptation, a biological story written over hundreds of thousands of years. Modern science has learned to read this story with extraordinary clarity. Through genetic ancestry analysis, researchers can trace how groups of people spread across the continents, how ancient populations met and mixed, and how each of us carries the echoes of countless forebears. What once was myth and oral tradition now finds confirmation in molecular evidence, uniting genealogy with genetics. The foundation of ancestry testing lies in genetic variation, the small differences in DNA sequence that distinguish one person from another. While the majority of the human genome is identical across all individuals, about 0.1% varies. These variations, often single nucleotide polymorphisms or SNPs, act like genetic signposts. Certain SNP patterns are more common in specific geographic regions or lineages, and by comparing a person's pattern to global databases, scientists can estimate ancestral origins. The result is not a fixed identity but a probability, a spectrum showing how a person's genetic makeup reflects humanity's shared history. Two particular types of DNA provide especially clear ancestral records. The Y chromosome, passed from father to son, traces paternal lines. The mitochondrial DNA, passed from mother to all her children, traces maternal lines. Together they create a molecular lineage that reaches deep into prehistory. By comparing mutations that accumulate slowly over generations, scientists construct haplogroups, families of related lineages, and map how they spread over time. These analyses show that all modern humans descend from populations that originated in Africa roughly 200,000 years ago. From there, small bands migrated outward, carrying the same genetic heritage that now resides in every living person. Advances in sequencing have turned ancestry testing into a personal experience. Millions of people have submitted saliva samples to commercial companies, receiving colorful reports of percentages and regions, a little Northern European, some East African, traces of indigenous American or Asian ancestry. Beyond curiosity, these analyses reveal the deep interconnectedness of humankind. Boundaries drawn by culture or geography dissolve under the microscope. What we call race, science reveals as variation within a single, shared family. Yet, as with all genetic knowledge, ancestry testing requires care in interpretation. The data show probabilities, not absolutes. A region labeled Mediterranean or South Asian reflects shared patterns of genetic variation, not a definitive homeland. Populations move, mix, and change. Identities formed by culture and language cannot be reduced to molecular codes. There are also questions of privacy and consent. Genetic databases, when shared or sold, may expose information not only about individuals but about entire families or ethnic groups. The story of ancestry is collective, and so is the responsibility to protect it. Beyond the personal, genetic ancestry has illuminated the migrations of ancient peoples. Analysis of ancient DNA from bones and teeth has reconstructed entire chapters of human prehistory, showing how Neanderthals and modern humans interbred, how farming spread from the Middle East to Europe, and how Pacific Islanders crossed vast oceans guided by stars and genes alike. These discoveries connect the present to the deep past, showing that every human alive today carries within them fragments of many ancient lives. Our genomes are living museums, containing artifacts of survival and encounter. Reflecting on this perspective changes how one sees belonging. Heritage is not limited by borders or names. It is a continuous flow, a thread of connection that stretches backward beyond memory. The bloodlines that shaped you also shaped the world. The face you see in the mirror is the latest expression of a story written by countless generations who endured, adapted, and loved. As you rest, let that thought bring comfort. The same code that sustains you also links you to every living being. You are not isolated in time. You are continuity made visible. Each breath you take echoes through an unbroken line, reaching back to the first humans who looked at the same night sky. The molecules in your body carry their courage, their curiosity, their endurance. And as you drift into sleep, those same molecules continue their quiet work, renewing, repairing, and remembering. Sleep, then, is not separation from life's story but participation in it, a pause between one heartbeat and the next in a chain that began long before you and will continue long after. For billions of years, life has followed the same biological rule. Information flows from existing organisms to their offspring. Every cell inherits DNA from a parent cell, every species from its ancestors. But in the past few decades, humanity has begun to challenge that ancient pattern. Scientists can now create synthetic DNA, designing sequences that never existed in nature and inserting them into cells to perform new functions. It is a breathtaking development, one that blurs the boundary between what is living and what is engineered. For the first time, life's code is not only read but written by human hands. The concept of artificial life began with the ability to synthesize small stretches of DNA in the laboratory. By combining nucleotides chemically, researchers could build short genes and study their effects. As technology improved, those short fragments grew longer, until entire genomes could be assembled from scratch. In 2010, scientists at the Craig Venter Institute created a synthetic genome of the bacterium Mycoplasma mycoides, inserted it into a related cell whose own DNA had been removed, and watched the cell come to life. The new organism, dubbed Cynthia, behaved like its natural counterpart, but carried a genome designed entirely on a computer. It was the first cell on Earth to have a fully artificial set of instructions. The implications were immense. Synthetic DNA allows researchers to test theories about the minimal requirements for life, how few genes are needed for a cell to survive and reproduce. It also provides a platform for engineering organisms that can perform specific tasks. Scientists have designed bacteria that digest oil spills, yeast that produce medicines or biofuels, and microbes that synthesize biodegradable plastics. By altering sequences or adding new pathways, they can make living factories more efficient, sustainable, and adaptable. What evolution accomplishes through chance, synthetic biology achieves through design. This ability, however, raises profound questions. If a genome can be written from scratch, what defines life? Does it begin when molecules assemble and start replicating, or only when they arise naturally? Philosophers and scientists alike have debated whether synthetic organisms are truly alive or simply complex machines made of biological parts. In practice, the distinction grows thin. Artificial or not, these cells metabolize, divide, and evolve. They obey the same chemical laws as all living things. In essence, they demonstrate that life is a process, not a substance, an organized flow of information and energy that can emerge wherever the right conditions exist. The creation of synthetic DNA also opens possibilities for innovation beyond biology. DNA's structure makes it an ideal storage medium for digital data. A few micrograms can encode entire libraries or archives, remaining stable for thousands of years. Scientists have stored text, images, even movies within strands of synthetic DNA, merging information technology with molecular biology. The code that once carried genes now carries knowledge itself, uniting two languages, binary and biological, into one. Still, the same tools that enable progress also demand caution. Synthetic organisms, if released without control, could disrupt ecosystems or transfer artificial genes to wild populations. Biosecurity concerns have led to strict regulations requiring that such life forms remain contained or designed with built-in limits, such as dependency on synthetic nutrients that prevent survival outside the lab. The challenge is to balance exploration with responsibility, invention with foresight. Perhaps the deeper meaning of synthetic DNA lies not in domination over nature, but in reflection upon it. The more we learn to build life, the more we understand its fragility and elegance. To construct a genome from the ground up is to glimpse the intricate logic that evolution refined over billions of years. The act itself becomes an acknowledgement of how finely tuned existence already is. If you imagine the scene, picture a scientist leaning over a glowing screen, assembling letters of DNA like a composer arranging notes. The sequence appears, strand by strand, until the cell awakens and begins to move, a spark of chemistry turned self-sustaining. It is not creation in the divine sense, but continuation, another verse in life's expanding symphony. The melody is older than humankind, yet now, through knowledge, we have learned to hum along. Every living organism leaves a molecular footprint. With every step, breath, or touch, fragments of DNA drift into the world. Cells shed from skin, pollen scattered by wind, plankton dissolved in ocean spray. These traces form an invisible network known as environmental DNA, or eDNA, a record of life's constant presence and change. Unlike fossils or bones, which take ages to form, environmental DNA is immediate and dynamic. It captures a snapshot of existence in the present moment, allowing scientists to study ecosystems without ever seeing the organisms themselves. The principle behind eDNA is simple. Every living being releases genetic material into its surroundings. In water, this material can linger for hours or days before degrading. In soil, it can persist for years. By collecting and analyzing samples from the environment, researchers can identify the species that have passed through, feeding, migrating, or breeding. A liter of river water may contain the DNA signatures of hundreds of species, fish, amphibians, insects, and even mammals that came to drink. A spoonful of soil from a forest floor might reveal the genetic traces of roots, fungi, bacteria, and animals alike. This technique has transformed ecology. Traditionally, studying biodiversity required capturing or observing organisms directly, an approach limited by time, visibility, and accessibility. eDNA sampling, by contrast, allows for non-invasive monitoring across vast areas. Scientists use it to track endangered species, detect invasive ones before they spread, and survey entire ecosystems after environmental change. A few drops of melted snow from a mountain stream can tell whether a rare frog still survives there. Airborne DNA sampling now identifies birds and insects as they fly past, turning the atmosphere itself into a biological archive. The applications extend far beyond conservation. Marine biologists use eDNA to map coral reef health, measuring how pollution or warming oceans alter community composition. Epidemiologists monitor wastewater for traces of pathogens, tracking disease outbreaks with molecular precision. Archaeologists extract ancient eDNA from sediments in caves or permafrost, reconstructing long-lost ecosystems without a single fossil. Each sequence recovered is a voice from the past, whispering who lived and how the environment once thrived. What makes eDNA remarkable is its universality. The same molecule that defines an individual also connects it to the environment. Every breath you exhale, every hair that falls, contributes to this planetary exchange. Life and surroundings are in constant dialogue, each shaping and recording the other. The biosphere, viewed through the lens of eDNA, becomes a living memory, a system where nothing is truly isolated and where even absence leaves a trace. Yet the power of eDNA also carries challenges. Its sensitivity means that contamination is easy. A single misplaced cell from a researcher can distort an entire dataset. Interpretation requires care, for detection does not always mean presence. A species' DNA might travel far from its origin through wind, water, or predators' waste. The science of environmental genomics is still refining its accuracy, balancing excitement with rigor. But even in its imperfections, it reveals a deeper truth. Life is not confined to organisms, but diffused across the spaces they inhabit. In a broader sense, environmental DNA blurs the line between individuality and community. It shows that existence is shared, that every creature contributes to a collective signature of life. The forest, the ocean, the sky, and even the human city each hold molecular echoes of their inhabitants. To study them is to read the Earth's current diary, written not in ink but in nucleotides. If you imagine the world at night, beneath still waters and quiet air, countless fragments of DNA drift and settle like invisible dust. Together they form a silent chorus, proof that even when unseen, life endures in every grain and droplet. The planet remembers us all, not in monuments or words, but in the gentle chemistry we leave behind. In this way, the Earth itself is a living genome, endlessly rewritten, endlessly aware. At its deepest level, life can be understood not only as chemistry but as information. DNA is more than a molecule. It is a data system, one that encodes, transmits, and corrects information with remarkable precision. Every strand of DNA contains instructions written in a 4-letter alphabet, and every cell in every organism carries the machinery to read, copy, and interpret those instructions. When viewed through the lens of information theory, first developed in the mid-20th century by mathematician Claude Shannon, the principles that govern data transmission also describe the behavior of living systems. Life, in this sense, is the universe's most enduring communication network. Shannon's theory defined information as the reduction of uncertainty, the amount of choice eliminated when one message is received instead of another. In digital systems, information is measured in bits. In biological systems, it is measured in bases. The genome's sequence of A, T, C, and G represents an immense stream of data, each nucleotide contributing roughly 2 bits of information. A single human genome, with 3 billion bases, carries about 6 billion bits, nearly a gigabyte of digital content. But unlike a computer file, this data is self-replicating, self-correcting, and subject to interpretation. Meaning in DNA arises not only from sequence, but from context, timing, and interaction. The parallels between digital and biological communication run deep. Just as electronic messages can be corrupted by noise, DNA replication can be disrupted by mutation. Cells employ error-correcting codes, mechanisms that detect and repair mistakes, analogous to redundancy in digital systems. Multiple codons can encode the same amino acid, ensuring that some mutations have no effect on the resulting protein. This redundancy is nature's safeguard against the noise of molecular life. Evolution, in effect, has optimized the genome to balance efficiency with resilience. But the comparison goes beyond mechanics. Information theory helps explain why life evolves complexity. In any system, information tends to increase when energy is available to sustain it. The flow of sunlight through the biosphere drives organization, allowing DNA to store ever more intricate instructions. Entropy, the natural tendency toward disorder, pushes systems to decay, yet life continually pushes back by importing energy and maintaining structure. This constant exchange between order and chaos is what defines living systems. Islands of information in a sea of entropy. When scientists measure information content in genomes, they discover that it correlates not simply with organism size or intelligence, but with evolutionary strategy. Some viruses, with only a few thousand bases, compress essential information into minimal genomes. Humans, by contrast, carry vast non-coding regions that help regulate complexity. In both cases, efficiency and adaptability arise from how information is organized, not how much exists. The genome is not a hard drive, but a dynamic network, storing patterns that can change with experience and environment. Information theory also bridges biology with physics. At the quantum level, the universe itself may operate as a computational system, every interaction exchanging bits of information. DNA participates in this cosmic dialogue, translating raw physical energy into living form. The transition from chemistry to biology was, in this view, a leap in information density, a way for matter to begin remembering itself. Each living being, by replicating and evolving, extends that memory, turning existence into a continuous act of communication between past and future. From this perspective, life's meaning can be seen as information persisting against oblivion. The genes you carry are not random strings but refined messages, successful solutions tested by countless ancestors. Their survival means they worked, that they made life possible. When you breathe, eat, think, and move, you are expressing information that originated in ancient seas. Your body is a pattern written long ago, still being edited by experience and environment. If you close your eyes and imagine the earth from afar, it appears silent, yet in truth, it hums with the exchange of data, cells reading, copying, signaling, and responding. DNA is the language of this planet, spoken fluently by every organism. It is the alphabet of persistence, the grammar of adaptation, the syntax of survival. Through it, the universe has found a way to describe itself, and through you, it continues the conversation. For most of Earth's history, DNA served a single purpose: to preserve the instructions of life. Yet in recent years, scientists have learned to use this molecule in a completely new way, as a storage medium for digital information. The same structure that carries the blueprint for cells can also store books, photographs, music, and even films. This fusion of biology and technology redefines what memory can mean. In the double helix, nature has provided the most compact, stable, and energy-efficient data system ever discovered. Humanity has begun to borrow that design, turning the language of life into the language of knowledge. The idea began with a simple observation. DNA encodes information using 4 symbols, A, T, C, and G, while computers use 2, 0, and 1. By translating binary code into base sequences, digital data can be written into synthetic DNA strands. These strands can then be stored in vials or dried on glass, taking up almost no space. A single gram of DNA, in theory, can hold over 200 petabytes of data, the equivalent of millions of modern hard drives. Unlike magnetic or optical media, which degrade within decades, DNA remains stable for tens of thousands of years if kept cool and dry. In terms of density and longevity, it far surpasses any human invention. Early experiments demonstrated this potential dramatically. In 2012, researchers encoded a digital version of a book, complete with images, into DNA and successfully retrieved it without error. Soon after, entire operating systems, films, and works of art were encoded as well. Today, companies and research institutions are developing automated systems that convert digital files into DNA sequences and back again, promising storage devices that could last as long as civilization itself. The double helix, once confined to living cells, now holds the words of poets, the images of galaxies, and the archives of human history. The beauty of this technology lies in its unity with biology. The same molecule that stores the story of evolution now preserves the story of culture. In one sense, it is humanity's attempt to achieve what life has always done, to resist entropy by preserving information. In another, it is a symbolic reunion of knowledge and nature. The archives of humanity now mirror the code of life itself, bridging organic and artificial memory. Yet this innovation raises philosophical questions as profound as its technical ones. When human creations reside in DNA, where does biology end and technology begin? Could these synthetic sequences one day interact with living cells or even influence them? The distinction between natural and artificial grows less clear. Some scientists suggest that future systems might use living cells as data centers, engineered microbes that store and retrieve information biologically. The boundaries between life and machine blur into a continuum of computation. Practical challenges remain. Synthesizing and reading DNA are still slow and expensive processes, far from replacing conventional storage for everyday use. Error correction and retrieval efficiency must improve before the technology can scale. Yet, progress is steady, and the long-term vision is compelling, an archive that could endure for millennia without power, maintenance, or obsolescence. If humanity ever leaves Earth, DNA could carry its libraries among the stars, a molecular time capsule of thought. Reflecting on this transformation reveals a deeper symmetry. Nature invented DNA to remember itself. Humans now use it to remember their world. In doing so, we echo the same impulse that shaped life, the desire to preserve meaning against the passage of time. If you imagine a quiet vault deep beneath the surface of the earth, shelves lined not with books but with tiny glass vials, each containing clear liquid. Inside those vials rest sequences of A, T, C, and G that encode the sum of human experience, language, art, science, history. No screens glow there, no machines hum. Only silent molecules hold what we once called civilization. Long after metal corrodes and paper fades, the double helix may still whisper our story to whatever future minds arise to read it. DNA, the molecule of life, may yet become the molecule of memory, proof that information, once born, never truly disappears. It simply finds new forms in which to endure. For billions of years, evolution unfolded without intention. Random mutation and natural selection shaped every living form, from the simplest microbe to the most complex brain. Now, for the first time in Earth's history, one species has gained the power to influence that process consciously. Humanity stands at the threshold of directed evolution, a new era in which genetic change is not left to chance, but guided by design. The future of life may no longer be written solely by nature, but by the choices we make. Already, the tools of this transformation are in motion. CRISPR and other gene editing technologies allow scientists to rewrite genomes with precision. Synthetic biology lets them design entirely new organisms, capable of producing fuels, medicines, or materials unknown in nature. Cloning can recreate individuals from preserved DNA, blurring the line between death and return. And gene drives, which force specific traits to spread rapidly through populations, could eliminate diseases carried by mosquitoes or control invasive species that threaten ecosystems. These capabilities mark the beginning of a new evolutionary phase, one driven not by slow adaptation, but by intention and innovation. The potential benefits are extraordinary. Genetic editing could eradicate hereditary illnesses that have plagued families for generations. Crops could be engineered to thrive in poor soil or changing climates, ensuring food security in a warming world. Endangered species might be revived or strengthened, restoring balance to fragile habitats. Even aging itself, the gradual accumulation of cellular damage, might one day be slowed or reversed through repair of DNA and control of gene expression. Humanity's relationship with biology is shifting from that of passive participant to active collaborator. Yet with this new power comes profound uncertainty. Evolution is not merely the sum of genes, it is an intricate interplay between organisms, environment, and time. Intervening in that balance carries risks we cannot fully predict. A gene drive that eliminates a pest could also disrupt an ecosystem. A modified organism could spread in ways its creators never intended. What begins as correction could become domination, replacing diversity with uniformity. Nature's resilience depends on variation, on the very unpredictability that editing seeks to control. Ethical questions, too, expand with possibility. Who decides what constitutes improvement? How far should human influence extend over the genome of other species or our own? The same technologies that can heal could also enhance, giving rise to engineered abilities or traits. If evolution once shaped humanity, humanity may now shape evolution in return. The boundary between medicine and modification, between care and control, may blur beyond recognition. Some philosophers argue that this moment marks a transition in evolution itself, the point at which intelligence becomes the new selective force. Natural selection, once blind, now gains awareness through human minds. Others caution that self-directed evolution could be an illusion, that complexity arises from collective interaction, not design, and that control may prove far more difficult than imagination allows. The genome is not a machine to be programmed. It is an ecosystem of relationships, one we are only beginning to understand. Still, the impulse to improve life, to alleviate suffering, to extend vitality, to explore possibility, will not fade. The challenge will be to act with humility, to recognize that every modification carries unseen consequences. Evolution, even when guided, should remain an act of respect for the processes that made it possible. The most ethical path forward may be not to dominate nature, but to partner with it, learning to adapt alongside it rather than above it. If you imagine the Earth centuries from now, you might see a landscape where natural and designed life coexist. Forests seeded with resilient species, oceans replenished by engineered algae, and humans whose health and longevity reflect the wisdom of carefully balanced science. The blueprint of life will continue to evolve, but the rhythm of change, discovery, adaptation, renewal will remain the same. The future of evolution, then, is not a departure from nature, but its continuation in a new key. Life's central rule endures. Information adapts to survive. Whether through mutation or design, the genome will keep changing, exploring what it means to exist. And as it does, it will carry with it a quiet reminder that every act of creation, no matter how advanced, still begins with the same four letters, rearranged into endless forms most beautiful. From the smallest cell to the vast expanse of humanity, one truth endures: life continues. DNA, the molecule that began as a fragile thread in ancient seas, has traveled unbroken across billions of years, passing through every creature that has ever lived. Its letters have copied themselves through storms, extinctions, and endless generations, carrying the same essential instruction: to persist. All that you are, every heartbeat, every thought, is the latest expression of that ancient continuity. The molecule within you has never truly died. It has only changed form, finding new ways to remember existence. When you look at life through this lens, Individuality softens. The DNA that defines you is not solely yours. Half came from your mother, half from your father, but all of it descends from lineages far older than memory. You are the current chapter in a single, continuous book written by the earth itself. The sequences in your cells were once carried by microscopic organisms that floated in primordial oceans, by plants that turned sunlight into energy, by animals that walked, flew, or swam long before you existed. Through them, the molecule learned resilience, invention, and connection, lessons still written into your body today. Every breath continues the same exchange that began when the first living cells learned to trade gases with their surroundings. Every cell division in your tissues repeats the act that first separated one organism into two. The genome has never stopped practicing the art of renewal. Its strength lies not in perfection, but in persistence, the ability to survive damage, to adapt, to grow from what remains. When you think of life as a continuous flow of information, even death becomes part of its motion. The molecules released by one generation are recycled by the next. Atoms that once belonged to ancient organisms now reside in you, and one day will move onward again. Nothing truly vanishes. It transforms. Modern science shows that this continuity extends far beyond Earth. The building blocks of DNA, adenine, guanine, and other organic molecules have been found in meteorites and in the cold clouds of interstellar space. It seems the universe was fertile with possibility long before life began here. From cosmic dust to cell, the pattern of replication was waiting to emerge. In this sense, DNA is not only a record of Earth's history, but a local expression of a universal principle, the drive toward structure, memory, and awareness. The cosmos itself has learned to reflect upon its origins through you. If you imagine this continuity, it may bring a sense of calm. The struggles and changes of life, seen across such a vast timeline, become gentle ripples on an endless current. Your existence, though fleeting in time, is permanent in connection. The DNA within you will one day return to the cycle, through children, through nature, through the exchange of matter that never ceases. In this way, you are part of a story that has no beginning and no end, a narrative written in 4 letters and spoken by all living things. As you drift into sleep, remember that your body is still part of that story. Within you, cells continue their quiet work, copying, repairing, and renewing. The same code that guided ancient life continues to hum beneath your awareness, steady and sure. While your mind rests, your cells write another chapter, one that will carry forward long after tonight. The continuity of life is not something you must hold on to, it holds on to you. It has done so since the dawn of time and will continue long after the stars themselves have changed. So rest, knowing that the thread of DNA, fragile, enduring, luminous, connects you to every form of life that ever was and ever will be. In that connection lies peace, purpose, and belonging. We from Sleepy Stories wish you a good night.